From the days of the earliest machines, we have relied on rigidity, levers, linkages, shafts and screws, connected to one another in intricate testaments to human ingenuity. However, if we look closely at the natural world, we see something else entirely. Fins, flippers, tails, tentacles, all bending and swaying together in a carefully choreographed dance. This graceful ballet is guided in part by hydrodynamics, the invisible, intricate interactions of the eddies and swirls created as each surface moves back and forth in the water.
My name is Margaret Byron, and I'm a 2021 Beckman Young investigator. I study environmental fluid dynamics, which is how air and water move in the environment. And I'm curious about how that can impact problems in both biology and in engineering. Thanks to the Arnold and Mabel Beckman Foundation, we are investigating how flexible biological propulsors can interact with each other to generate the fluid flows that animals use to swim. We develop new tools for both engineers and biologists to visualize and study these flows so that we can really understand the physics behind the incredible performance and efficiency that we see across the animal kingdom. And with that knowledge, we can build the next generation of bio-inspired vehicles, devices, and robots. While lots of animals are flexible, we're focusing on comb jellies, or ctenophores. These are a type of sea jelly, and not only do they live in pretty much every ocean habitat, they're the largest animals in the world that use cilia to swim. They organize hundreds of thousands of these little hairs into paddle-like structures that beat in a coordinated pattern called a metaclonal wave. We use high-speed cameras and special imaging tools to measure these waves and the flows that they make, which help the ctenophores swim and catch their prey.
Hi, I'm Reza Jarfa, and I'm a PhD student studying the fluid mechanics of flexible biopropulsors. We've worked with these animals both in the field at Friday Harbor Labs in the San Juan Islands of the coast of the Washington state, as well as in the lab here at Penn State. We're looking at the way the rolls of the propulsors create momentum in the surrounding fluid, and how the vortices they generate can interact with each other to generate propulsive forces for swimming.
Another reason we're studying flexibility in animal swimming is to help develop the next generation of bio-inspired underwater robots. There are a lot of really great new tools in soft robotics and materials that we can use to explore all kinds of possibilities.
Hello, my name is David Peterman. I'm currently a postdoctoral scholar here at Penn State, and we're currently exploring different ways to investigate flexible biopropulsors using magnetic elastomers. These magnetic elastomers are made out of neodymium microparticles suspended in a flexible silicone matrix. We can take these samples and put them in a strong magnetic field to encode specific beating patterns in the propulsors. And then later they can be actuated with these translating magnets underneath. And these translating magnets produce a time-varying magnetic field where we can adjust beat frequency and phase lag and other properties of these metachronal propulsors. And we can use this approach in combination with flow visualization techniques to characterize how they generate fluid flow. We've also been using these magnetic elastomers to develop a fully autonomous ctenophore-inspired robot. And using this approach, we can coordinate hundreds of propulsors with a specific phase lag and beat frequency.
For me, the most striking thing about Arnold Beckman is just his dogged pursuit of solutions whenever he came across an unsolved problem, and his willingness to stretch outside some predefined idea of who he was supposed to be or what his skill set was supposed to look like. Oh, so we don't have a way to measure that thing. Okay, let's figure out how to do that, right? Let's measure pH, let's measure chemical composition, let's measure oxygen levels, let's measure air pollution. His vision was almost limitless, up to and including funding the semiconductor labs that were the foundation of the original Silicon Valley. And you can see the impact of these technologies everywhere in so many areas of people's lives. It's something I personally aspire to as an engineer, to just build things that help people. I also just really appreciate that he never took himself too seriously and that he was devoted to his family and his kids, his spouse throughout his life. I was able to use some of my Beckman Young Investigator Award to support childcare for my 2 young children during one of our field seasons at Friday Harbor Labs, which let me stay longer and collect more data. So my kids got a front seat to some really amazing work being done by all my colleagues there. And most importantly, they got to see what it looks like for a mom with young kids to be actively pursuing a career as a scientist. I think this is really important for young people, that they form their conception of who gets to do science in the broadest way possible. This is a core value that I'm taking into my future as a leader in science and in engineering. This project really enabled us to kickstart our work on the idea of how flexibility can enhance propulsion in biological swimming systems. We're going to continue this work and extend it to other squishy swimmers as well, and continue to look at the incredible diversity of animals and their appendages and how we can use them as inspiration for new technology. We are so grateful to the Arnold and Mabel Beckman Foundation for making this work possible, and we can't wait to keep exploring.